Hello, good afternoon, and assalamu alaikum, everyone. I hope every one of you is doing good and safe and healthy at home. All right, today's session is going to be chapter 5 for our class and chapter 13 in our book, The Microbiology and Introduction by Tortora, Fung, and Case. All right, today's chapter is on the viruses and viroids. So we're just going to mention a little bit on the differences of what a virus is and a viroid is. Viruses are actually too small to be seen with a light microscope and can even be cultured outside the host cell in comparison with bacteria. Therefore, although viral diseases aren't new, they are quite old, the viruses themselves couldn't be studied until 20th century. In late 1800s, a Dutch chemist with the name of Adolf Meyer, he showed that the tobacco mosaic disease, or in other words, it's called as a TMG, it's a disease that was transmittable from plants to infected plants to healthy plants. And in 1892, in an attempt to isolate the cause of the TMD, the tobacco mosaic disease, another scientist, which was a Russian scientist, a bacteriologist in this time, his name Dmitry Ivanovsky, he filtered the sap of the disease plant through a porcelain filter that was designed to retain the bacteria. He expected to find some microbes, some bacteria in the filter, and instead he found that the infectious agent had passed through the minute pores of the filter. When he infected the healthy plants with the filtered fluid, he contracted TMD. He himself contracted it. So this was the first human disease which was associated with a filtered agent, in this case the virus. And it was known at that time as a yellow fever. I'm sure a lot of you have heard about it. Advances in molecular biology techniques in late 1880s and then later on in 1990s led to recognition of several new viruses. Until that time, it was very hard to identify the cause of a viral disease. So in late 1990s or early 1990s, one of the major outbreaks in the identification of virus came about, which was the human immunodeficiency virus, or HIV, and later on the SARS associated coronavirus, and nowadays the COVID-19. All right. Today's chapter, the learning outcome, there are a couple of learning outcomes. We will start with the general characteristics of the virus and how do we define the virus? That's the most important question that anyone would ask. So basically, viruses are originally distinguished from other infectious agents because they are especially small and, like I mentioned earlier, filterable agents, and because they are obligate intracellular parasites that is absolutely require living host cells in order to multiply. So the phrase itself, obligate intracellular parasite, obligate, which means compulsory, intracellular, which means inside the cell, and parasite means it depends on it. So they require living inside the host cell to multiply. They have to be inside the host cell in order to replicate itself, in order to live, in order to survive. If they are not inside the host cell, they cannot survive. So viruses, they do contain DNA or RNA. They are very simple in their structure. They have a protein which is coating the genetic material, the DNA or RNA. They have no ribosomes. They have no ATP generating mechanisms such as mitochondria in bacteria. And yes, they are very, very simple. However, both bacteria and virus, they share some certain similarities uh, with each other. Not with all bacteria, but there are certain bacteria such as Rickettsia and Chlamydias. As you can see in there, Rickettsia and Chlamydias, they are both intracellular parasites, same as virus, and they both pass through bacteriological filters. So meaning that they are very small, they are very tiny and small in size. And adding to it, um, some bacteria, such as chlamydia, they do not have ATP generating metabolism like the virus itself. So these are very few similarities, but this is just to make a comparison. However, when you compare with the rest of the bacteria, bacteria basically from the previous chapter, you have understood that they do generate energy themselves. They do have ribosomes. They do have both RNA and DNA. They can have in a single cell. And they do not pass through filters because they are bigger in size. And they have a different way of replicating itself, such as through binary fission. They have plasma membrane. Virus doesn't have it. And they are not considered intracellular parasite because they don't have to be inside the cell in order to survive. All right, host range. The host range of a virus actually is the spectrum of host cells the virus can infect. How many types of cells the virus can infect? There are viruses that infect invertebrates, vertebrates, plants, protists, fungi, and bacteria. So basically, any single cell that comes on their way, viruses are capable of infecting because they are the tiniest of all. However, most viruses are able to infect specific types of cells of only one host species. In rare cases, viruses cross the host species barrier, thus expanding their host range. So most viruses are only able to infect the cell of interest, if we can say in other words. Like for instance, we do have the viruses that infect only the bacteria. And those viruses are called bacteriophages. So bacteriophages are specific to their host, which is bacteria, and thus they are not able or capable of causing any infection in human cells. And when it comes also to the bacteria itself, there are specific bacteriophages for specific species of bacteria. And viruses are very, very small in size, which they range from 20 to 1000 nanometer in length. All right, here in this picture, we have the viruses range, which I just mentioned, and the comparative sizes of several viruses and the bacteria and human cell. You can see that E. coli, it's quite big. It's almost 3,000 times 1,000 nanometer inside, and chlamydia that we compared together with the virus just now, it's 300 nanometer, which basically can penetrate the pores when they are filtered. with a porcelain filter. All right. In here we have the viral structure, and the term that I mentioned to you is virion. So what actually is a virion? A virion, it's a complete, fully developed infectious viral particle, which is composed of a nucleic acid and surrounded by protein code outside the host cell. So basically, a complete structure of a virus is called a virion. Viruses, they are classified based on their nucleic acid and by differences in the structure of their host cell. So I mentioned earlier the nucleic acid can be DNA, RNA, and we have a single or double-stranded, linear or circular. In contrast with the prokaryotic and eukaryotic cells, in which DNA is always the primary genetic material, and RNA plays an auxiliary role, a virus can have either DNA or RNA, but it can never have both of them in one. So the capsid is basically the protein code which covers the genetic material, and it's made of small units or subunits, which we call them as capsomeres. So basically, capsomeres are small proteins which they join together and they make up a capsid. An envelope, it's made of lipids. Lipids, proteins, carbohydrates, which basically coats the capsid. So not all the viruses have envelope. We have envelope viruses, we have non-envelope viruses. And sometimes when the viruses are envelope viruses, they do have spikes or projections from the upper surface. Here we have a morphology of a non-envelope virus, which is polyhedral in shape. And you can see in there the nucleic and the surrounded by or coated by the capsid, and the capsid made of these small balls which are protein and are called capsomeres. That's the most basic structure of a virus. And here we have the morphology of envelope virus, and you can see in there it's similar to the non-envelope. The only difference is that it has another cover above the coating, and it's called an envelope, and it contains spikes. All right, and okay. So the general morphology of the virus, when you have seen under the bacteria chapter, bacteria comes under many different shapes, different sizes. Sometimes it's the arrangements also that matters when you identify. The general morphology of the virus is very, very simple. It comes in 4 different shapes. We call it as helical viruses, polyhedral viruses, envelope viruses, and complex viruses. So complex viruses, basically it's a joint structures of many different viruses in one, which makes one virus. It's not many different viruses, it's just like different morphologies in one single structure. All right. Here we have the morphology of a helical virus. You can see it's in a helical shape, and it only contains the capsid. It's a non-envelope virus. Here we have a very good example of a complex virus. We have the polyhedral, which is the head. It's the polyhedral shape. And then we have the sheath, which is comprised of helical shape and is joined in one, which basically is making a complex virus. And you can see the spikes as well, the tail fiber in there. And this particular virus, it's called bacteriophage, which is bacteria infecting virus. Or another type, which is also a complex virus, is this orthopox virus. Okay, taxonomy of viruses. Just as we need taxonomy categories for plants, animals, and bacteria, when we want to identify them and classify them, we need also viral taxonomy to help us organize and understand newly discovered organisms. The oldest classification of viruses is based on the symptomology. What it means is based on the disease that affects different systems in the body, and more specifically, in early days, it was um, for the diseases that affect the respiratory system. This system was convenient at that time, but not scientifically acceptable, because the same virus may cause more than one disease depending on the tissue affected. In addition to that, the system artificially grouped the viruses that don't infect humans. So it was very hard to classify the viruses based on the symptomology. Right, so viruses, they do not have a kingdom, they do not have a phylum, what we had mentioned earlier during the class sessions. Viruses are considered non-living organisms, despite what we, that's a class 10, and despite what we actually believe ourselves. So the virus taxonomy starts from the taxon, we call it as genus. So when you have a name of a virus, it ends with if it ends with virus, it's related to the genus. The family names end in viridia, and the order names, it ends in alis. So viral species, in other words, there are a group of viruses which share the same genetic information and ecological niche. So they share the same names, and common names are used for species, and subspecies are designated by a number. What do I mean that? For instance, we have like the fact that the viruses can multiply outside a living host cell complicates their detection, enumeration, and identification. So viruses might be provided with a living cell instead of a fairly simple chemical medium. Living plants and animals are difficult and expensive to maintain, and pathogenic viruses that grow only in higher primates and human hosts cause additional complications. However, viruses that use bacterial cell as hosts, like bacteriophage, are rather easier to grow on bacterial culture, and this is one of the reasons so much of our understanding of viral multiplication has come from bacteriophage. So bacteriophages grow in bacteria, and when they grow in bacteria, they do form some plaques, which are a clear zone on a lawn of bacteria on the surface of agar, and each plaque corresponds of a single virus, which can be expressed as the plaque forming unit. And when in bacteria, we call it as a colony forming unit. Here in this picture is you can see the plaques, the clear zones. So each plaque corresponds to a single virus that can be expressed as a plaque forming unit, or PFU. So basically, based on the amount of the plaques on the plate, you can calculate it. You can calculate the PFU of the phage, the infected phage. Okay, growing animal viruses in the laboratory. In the laboratory, three methods are most commonly used. for culturing animal viruses. When we say animal viruses, it's viruses which basically infect human cells or animal cells. And these three methods involve using living animals, embryonated eggs, or cell culture. So these are the three most common techniques which are used to grow the virus in the lab. All right, so this picture in here is inoculation of an embryonated egg. So you use needle, you have to poke the needle in there, and the viruses will grow on the membrane at the inoculation site. You can see in there the inoculation site of the virus in the egg. So there are many different spots inside the egg which you inoculate the virus. Okay, cell culture have replaced the embryonated eggs as a preferred type of growth medium for many viruses. It consists of cell growth, cells that are grown in cultured media in the laboratory. And because these cultures are generally rather homogeneous collections of cells and can be propagated and handled much like bacteria culture, they are more convenient to work with than the whole animals or embryonated eggs. They are very easily handled. You need to change the media of the cells daily. So basically every 24 hours you need to maintain their pH and temperature and the cleanliness of the media. So you have to make sure that the media itself is not contaminated with other microbes such as bacteria, and it's much easily handled compared to the embryonated eggs. Here is a diagram of the cell culture. So the transformed cells can be grown indefinitely in laboratory culture. The first thing which you have to do is a tissue is treated with enzymes to separate the cells, and we're talking about the animal tissue. And then second step is the cells are suspended in the cultured media, which is nutritionist media, contains nutritional values that the cells can grow or helps the cells to grow. And then we have like normal cells are primary cells grown in monolayers across a glass or plastic container. And transformed cells or continuous cell cultures do not grow in monolayers. All right, here we have a picture of the cytopathic effect of viruses. In picture A, you have the uninfected human cervical cells, and you can see in there each one of them has one nucleus. In picture B, you have after inoculation. So they were inoculated with HHV virus 2, and the red cervical cell, which you see in there, has many nuclei that are filled with viruses. All right, so when it comes to viral identification, how to identify the viruses, which are the techniques, which I'm sure every one of you is wondering, because right now we are in a crisis of a virus outbreak out there, and there are every single day or throughout the day there are reports on, okay, this patient was identified with the virus, was positive, was negative, the results were not taken yet. So I'm sure each one of you must have been wondering, it's like, what are the techniques which they are using in order to identify these viruses? What samples are they taking from patients? Well, actually, when it comes to samples, that's a totally different story, because depending on the disease itself, whether if the virus is causing a respiratory disease, then you have to take a sputum, you have to take a saliva, you have to take a mouth sample, and etc. in order to identify the virus. But we're talking about the techniques in here. So identifying a viral isolate is not that easy. It's not an easy task compared to the bacteria. For one thing, viruses can be seen without the use of an electron microscope. We cannot use a compound microscope like we use in our lab. So we have the serological techniques or methods. We have the Western blotting, which is part of serological test, and these are the most commonly used means of identification. And Western blotting, adding to it also the ELISA assay, they mostly deal with identifying the antibodies which are found present in the blood sample. Because serological, it means sera, which is related to blood. It's a blood collection. And another technique which is very commonly used, or method, it's on identifying the nucleic acid of the virus. Now you got hold of the virus. Now you need to see what type of virus it is. You go deep into the viral structure of the virus itself. So there are two different methods. One of them is called restriction fragment length polymorphism, or RFLP. And the second one, which is very commonly used, is PCR, which stands for, I'm sure every one of you knows, polymerase chain reaction. So polymerase or PCR was used to amplify viral RNAs to identify for the first time, the Western Nile virus. I'm not sure if you guys have heard of it, in 1999 in the US. And then later on played a very crucial role in identifying something which is very common with COVID, the SARS virus, the SARS corona-associated virus in China in 2002. And PCR as well, giving you latest updates, is one of the methods which is being used to identify the COVID sample throughout the world right now. Okay, how does a virus multiply itself? The nucleic acid in a virion contains only few of the genes needed for synthesis of a new virus. So it only has few genes which it needs in order to make a new virus. And these genes include they include genes for the virion structure components, and those are, for instance, the capsid protein and genes for a few of enzymes which are used in the viral structure. What does that mean? I'm going to explain to you in a while. It's a little bit complicated unless it's shown. I'll give you a picture in a while. And these enzymes are synthesized and functional only when the virus is with the host cell. They do not work unless they are inside the host cell. So it must invade the host cell. All right. Here we have one-step growth curve, which shows, I'm sure some of you are already familiar with it because it's a hot talk right now in the media, on the news, bringing down or lowering down the curve. That's what they're talking about. They're talking about the curve, the release of virus from the host. They want to reduce that. They want to make it less spreading. So no new infective variants are found in a culture until after biosynthesis and maturation, meaning like until the proteins and the enzymes are synthesized inside the host cell, and the newly produced virus maturates or becomes completely assembled inside the cells, have taken place. So there is no infection unless the virus multiplied itself and then assembled itself and then has maturated into a grown-up virus. The infection won't happen. Most of infected cells, they die as a result of infection. Consequently, new variants won't be produced. So when the cell itself is dying while the virus are inside, there are new cells, new variants not produced. So in order for you to tackle the virus not being produced, sometimes you have to actually destroy the host cell. So the variant released from the host cell, it's reduced in number. And that's why once the viral release from the host cell is reduced in number, you have a decrease in the curve, the growth curve of the virus with the passing of time. All right, so we came to multiplication of virus, their life cycle. How do they actually, what is their life cycle? How do they multiply itself? Is it the same, the DNA and RNA viruses growing in the same way, or is it different? We have two different, we're going to start from the multiplication of bacteriophages. Okay, although the means by which a virus enters and exists a host cell may vary, the basic mechanism of a viral multiplication is similar to all viruses. Why we're choosing the bacteriophages? Because bacteriophages can multiply by two alternative mechanisms. We call it as a lactic cycle and lysogenic cycle. What it means is that in lactic cycle, ends with the cell ends with lysis and death of the host cell, whereas the host cell remains alive in lysogenic cycle. And what happens is the phage DNA, it's incorporated in the DNA of the host cell. Right? And there we have the specialized transduction. What it means, we're going to see next in a while. So you can see in here, I've put up there the T-even bacteriophages. The T-even bacteriophages are a certain type of bacteriophage. We have T2, T4, and T6. They have been studied most extensively, and they are well described. And the multiplication of the T-even bacteriophages in the host cell, which means bacteria cell, and mostly is studied in E. coli, it's an example of a lactic cycle. So this T-even bacteriophages, their cycle, it's a lactic cycle. So the first thing that they do when they want to enter inside a host cell is they attach themselves on the surface with their tail fibers, like what you saw, like a rocket kind of shape, and they penetrate inside. Or penetration is the second step. Like phage lysosome opens the cell wall, the tail sheath contract to force the tail core and DNA into the cell. It's something like a mother giving birth, where only the baby goes inside the cell. In this case, we're talking about the genetic material. The third step is the biosynthesis, which we have the production of phage DNA and proteins. The fourth one is maturation that I mentioned to you earlier, which we have the assembly of the phage particles. And the fifth step is release, which phage lysosome breaks the wall, even bacteriophage. And the reason why we're using that is that the bacteriophage lambda, it's a well-studied lysogenic phage, and it's a very good example of a lysogenic life cycle. So the lysogenic phage results in phage conversion, which means the whole cell exhibit new properties. The reason that it enters the DNA of the whole cell, the cell itself it's called a prophage cell now. The cell itself, the host cell, will start exhibiting some new properties, like starts functioning differently. in comparison. Time we have dissemblance of phage DNA from the DNA of the host cell, and we have back to the second phage, the second stage of the phage life cycle, where it's um, like the penetration, and then we have the biosynthesis and the lysis of the cell. So this for the prophage or lysogenic cells, the lysogenic bacteriophage, to lyse a cell, it might take a long time. Might stay in the body for a very long time, and until it's due to favorable conditions, and it can lead to lysing of the cell. Okay, specialized transduction that I mentioned earlier, there are specific bacterial genes which are transferred to another bacteria via the phage. That's called transduction, and which basically will lead to genetic changes or changes in the genetics properties of the bacteria. Okay, this is the specialized transduction, which basically it's a mixture of the genetic material of phage and bacteria together. So once the phage genome is disassembled from the genetic material of bacteria cell, it can carry along certain parts, certain genes of the bacteria as well. All right, the multiplication of animal viruses. Now we were just talking about the bacteria viruses. Now the multiplication of animal viruses follows a basic pattern of bacteriophage multiplication, but has several differences. Like what you can see in here, attachment, virus attached to the membrane cell. They enter by receptor-mediated endocytosis or fusion, meaning like the membrane of the cell will kind of like engulf the virus inside the cell. Uncoating by viral or host enzyme, and biosynthesis, which means production of nucleic acid and protein. It happens inside the cell. Maturation, also nucleic acid and capsid proteins are assembled, and we have the release by budding or envelope viruses or rupture, by rupturing of the host cell. There were three that we mentioned just now. One is receptor mediated, the A1, the A picture, receptor mediated endocytosis. The other one is by fusion, and the third one is by budding process. What you can see in picture A in there, it's a diagram of the budding process. And in picture B, we have a HIV budding from a T cell. This is a T cell in here, and we have a couple of HIVs. If you can notice, there are 4 budding HIV viruses which acquire their coating. You can see this is the coating part from the host plasma membrane. And a T cell, for you who do not know that, T cell is a type of lymphocyte, which is an immune cell, which develops in the thymus gland, and that's where it gets the name T cell, from the thymus cell, and plays a central role in the immune system, especially among the HIV infected patients. Okay, biosynthesis of DNA viruses. So now we're talking about how the DNA viruses are synthesized. We have RNA viruses, we have the DNA viruses. Now we're talking about the DNA viruses, their life cycle. When it comes to the DNA viruses, generally, the DNA containing viruses replicate their DNA. Remember this, they replicate their DNA inside the nucleus of the host cell. Inside the nucleus of the host cell. by using the viral enzymes, and they synthesize their capsid, those proteins which forms their coating and other proteins in the cytoplasm by using whole cell enzymes. Then the protein migrate into the nucleus and are joined with newly synthesized DNA to form virions, meaning like a complete virus. And these virions are transported along the endoplasmic reticulum to the host cell's membrane for release. So this is what I was talking about. So basically the attachment and entering, we have the same. However, uncoating of the virus is done inside cell. Okay, you can follow the structure behind here. So there are two key concepts in there. Viral replication in animals generally follows the steps what we just mentioned: attachment, entry, uncoating by synthesis of nucleic acid and protein, maturation, and release. Knowledge of viral replication phases is important for drug development strategies. So once you understand the viral replication, you are able to tackle it easier by producing the drugs which can tackle those concepts, and is good for the understanding the disease. The multiplication, however, of RNA viruses is essentially the same as that of DNA viruses, except that RNA viruses, they multiply in the host cell's cytoplasm. Like I mentioned to you, the DNA only multiplies inside the nucleus of the host cell. RNA multiplies in the cytoplasm of the host cell. There are several different mRNAs formation mechanisms that occur among different groups of RNA viruses. Okay, what that means, I'm going to explain in a while. So major differences among the multiplication processes lie in how that mRNA that is produced and viral RNA are produced. So these viruses have something which they need in order to replicate the virus. So there is an enzyme which we call it as RNA dependent and RNA polymerase. All right, that's confusing a little bit. Okay, so when it comes to RNA itself, RNA comes in two different forms. So one virus, if it's an RNA virus, it's only going to have one of these three forms of the RNA. So we have positive single sense, single-stranded RNA. We have negative single-stranded RNA, and then we have double-stranded RNA. Okay, so the viral RNA, which basically it's a positive single-stranded RNA, it serves, the RNA itself serves as an mRNA or messenger RNA in order to produce the protein. However, a negative single-stranded RNA, the viral RNA is transcribed, meaning that it's converted to a positive stranded mRNA for protein synthesis. And in order to do that, it needs an enzyme, and that enzyme is called RDRP, or RNase polymerase RNA dependent enzyme. Okay, now we have three different structures of RNA for RNA viruses. So each one of them, I'm explaining it differently. So what you see in this picture It's the pathway of multiplication used by various RNA containing viruses, and specifically this picture is on okay, single-stranded. This is a single-stranded RNA virus, which is a positive. It has a positive strand genome, which means positive single-stranded RNA are able to synthesize the protein directly. What I said, directly from their positive strand, and you can see here. Okay, this strand, okay, it enters inside and it automatically, it's basically synthesizing the DNA. So the DNA is straight away, it's synthesized in the okay, the genome are the proteins are synthesized from the positive strands. Using the positive strand as a template, they describe the negative strands, which means they transcribe the negative strands in here to produce additional positive strands to serve as mRNA and to be incorporated into capsid protein as the viral genome. So this part in blue here is basically what differs in the pathway of positive and negative single-stranded RNA. So the single-stranded RNA viruses with a negative strand genome, previously we had a positive strand genome, this one is with a negative strand genome, they must transcribe a positive strand to serve as mRNA. They must transcribe the positive strand in order to serve as an mRNA before they begin synthesizing protein. So the first thing that happens is not the protein synthesis, which we had in the positive stranded RNA. Here we have the transcription of protein. Transcription process, it means like when the protein is it's synthesized first. Okay, the mRNA is transcribes additional negative stranded for incorporation into the capsid protein. I'm sure is it clear, guys? I'm going to provide additional videos on the life cycle of the virus so it can make it a little bit more clear. So the third step is double-stranded RNA viruses. They must use mRNA, which means the positive strand, in order to code the protein, including the capsid protein itself. So the mRNA is produced inside the capsid and released into the cytoplasm of the host cell. So once it's released, the RNA polymerase initiate production of negative strand. The mRNA and negative strand form a double-stranded RNA that is incorporated as a new viral genome. All right, so a lot of information on the life cycle of viruses. So you guys have to sit and try to comprehend and make sense of it. So in other words, you have to study it, not just listen to my talk. So biosynthesis of RNA viruses that use DNA. This group includes a type of viruses which we call them as retrovirus and oncogenic RNA viruses. So these viruses, one very good example of these viruses is from the family of Retroviridia. Retroviridia family, many of the viruses from this family, they infect vertebrates. And one of the genus of retrovirus, it's called lentavirus. And lentavirus, one of the species of lentavirus is HIV, human immunodeficiency virus. And now we do have many different subspecies, such as HIV-1, HIV-2, 3, 4, and 5, which basically cause a disease called AIDS. So that's the difference between HIV and AIDS. HIV is a virus, AIDS is a disease. So the retroviruses that cause cancer are called oncoviruses. I reiterate, the retroviruses which cause cancer are called oncoviruses. In here, they have a description. Like I mentioned, in the RNA viruses, we have the viruses which basically, some of them, they depend on the RNA, dependent RNA polymerase enzyme, or DRP. In retrovirus, what happens is, these are also RNA viruses. The HIV virus is an RNA virus. What happens is, once the virus enters inside the host cell, they will need an enzyme in order to help RNA to be converted into a DNA. So the virus, this reverse transcriptase enzyme, it will make many different copies of RNA, will convert them into a DNA, and then once it converts into DNA, the virus will get inside the host cell. It will transcribe the whole cell. The transcription happens inside the nucleus, and then once the transcription, there are multiple types. There are multiple numbers of DNA produced. They will be released into the cytoplasm, and they are converted back into an RNA. Once they're converted back into the RNA, you will have the reassemblance of the virus, and then you will have the virus leaving the host cell. Again, here there are families of viruses that affect humans. You can look into it. There are many different types of them for your understanding and for your learning. And okay, I think I'm going to stop here. We have also another part of the chapter, which is viruses and cancer. I think I'm going to make a new video on that, not to make it very, very long, which I will attach to it maybe later on today or in another day. So for time being, you check your understanding by answering this question. Describe the principal events of attachment, entry, uncoating, biosynthesis, maturation, and release of an enveloped DNA containing virus. DNA containing virus, not an RNA. So practice it, try to draw it on your paper, and then once you have done so, why don't you send me a picture or send it to the group, and let's see what you've done. All right, as for today, thank you very much. I hope you guys are not bored listening to this lecture, and hope you have learned something from them. Any questions, any comments, please leave it on the Google Classroom under chapter 5, where the video is going to be placed. So once again, thank you very much, and best of luck to all of you. Thank you.